reappeared, the nucleolus were reappeared, and the chromosomes were started to the condensation. But that condensation, to the uncoiling. The uncoiling where can be it can be seen in case of the mitosis, because the complete mitosis occurs, the two daughter cells were formed. But here only the nuclear envelope you can see, or the nucleolus where may be appeared, because most of the time you have seen in case of the telophase, what happens? It simply started to form this because the cell has to go into the next phase also. So sometimes the cell plate formation occurs, or the division of the cell occurs, but sometimes it is not going to be happen. Simply the cell undergo the next divisional phase. So haploid cells of the chromosomes arrive at the respective poles. As I said in the previous class, when we were discussing about the metaphase stage, sister chromatids were attached. Only the homologous chromosomes, they were separated. Okay. So after the separation, the one set of the chromosome moved towards the one pole, and another set of the chromosomes were moved towards the another pole. But the chromosomes number become half. Then endoplasmic reticulum from the nuclear envelope around the chromosome, and they uncoiled. As you know that endoplasmic reticulum, how it is attached to the nuclear envelope. That we will discuss in detail when we were discussing about the nucleolus and the nuclear envelope. The nucleolus reappears. The cytokinesis takes place by furrowing in animal cells and the cell plate formation in case of the plant cells. And the karyokinesis and the cytokinesis resulted in the two haploid cells which undergo the short resting interphase, very short. But in some cases, you can find that the cytokinesis may not occur in some cases, or during the course of the division. Sometime it may not happen. Directly, it undergo the next phase. So the no DNA replication occurs. This interval of the missing the S phase is called as the interkinesis, and the interphase resulted in the similar chromosome structure as this appeared to the telophase one. Some organisms go directly into the anaphase to the metaphase second, from anaphase to metaphase second, skip the telophase and prophase second. So as I said, that in most of the cases, that kind of things occurs, that the proper telophase occurs, and then the small interkinesis, or you can say that the interface is there after the telophase before going into the prophase second. But sometimes in some organisms, it is found, or you can say that that maybe the cytokinesis occurs, or the division of the cells may occur, but it is very short that nobody is going to see it. So that may happen, but on the basis of the observation of the different workers, it is found that right from the anaphase directly the cell move to the metaphase one and the prophase second. Here you can see that the two dark structures, these are the two cells. These are the two cells, and two dark structures on the nucleus, on the respective poles. Yeah, that is very clear. दो डार्क स्ट्रक्चर आपको दिखेंगे, डार्क सर्कुलर स्ट्रक्चर्स. जब आप एक्सपेरिमेंट कर रहे होंगे या फिर जब आप प्रैक्टिकल्स कर रहे होंगे, तो ये दो डिफरेंट सेल्स हैं और इनमें दोनों में बहुत क्लियर आपको ये दिख जाएंगे. दोनों तरफ आपको डार्क सर्कुलर स्ट्रक्चर्स लिखेंगे. सो दैट मीन्स कि उन्होंने जो जो क्रोमोसोम्स हैं वही स्टेन लेते हैं जब हम ये वाला एक्सपेरिमेंट कर रहे होते हैं. So this is the identification of the telophase fast. It is very clear. दो बस डार्क स्ट्रक्चर दिख रहे हैं, तो ये बिल्कुल आपको क्लियर होना चाहिए कि ये टीलोफेज सेकंड है. ओ सॉरी, टीलोफेज फर्स्ट है. कभी कभी जब आप एक्सपेरिमेंट कर रहे हो तो कभी ये मत सोचिएगा. कभी कभी क्या होता है, कंफ्यूजन हो जाता है, दो सेल अटैच कर गए, ओवरलैप कर गए, और लगता है कि ये टीलोफेज सेकंड है. और इस फिगर में भी आपको, इस स्लाइड में भी ये लग रहा होगा कि ये दो सेकंड है, चार बन रहे हैं. ये चार जो बन रहे हैं वो बिल्कुल डिफरेंट है. ये सेल बिल्कुल डिफरेंट है, ये सेल बिल्कुल डिफरेंट है. एंड इट इज वेरी मच इम्पोर्टेंट to understand all these things. That only you can understand while you are doing the practicals. But we were not able to do all these things in the laboratory. So that I can give you an idea how they looks like when you are going to perform or you are going to appear in the practicals. Now come to the meiosis second. This is also an important part in case of the prophase second. Again, it's a very short stage, and the chromosomes with two sister chromatids visible, thick and short. So it's nothing new whatsoever the chromosome we are present, whatsoever the number of the chromosome we are present in the metaphase stage, and how they were moved towards their respective poles. So whatsoever the number of chromosomes present in the telophase, they were found same as in case of the prophase. So nothing changed. No any kind of replication occurs. No any kind of addition or deletion or any kind of overlapping, or you can say the exchange of the genetic material occurs. Only the same chromosomes with sister chromatids, with attached sister chromatids were present in case of the prophase second. The nuclear membrane and nucleolus disappear. So in the late prophase second stage, the nucleolus and the nuclear membrane is going to disappear. And the centrioles divide into two, resulting in the production of the two pairs of the centrioles. So whenever we are talking about the centrioles of any bodies which were resulting of the spindle fiber or the microtubules, because in most of the you can see in case of the plant cells, that kind of structures were not found the centrioles. Some kind of microtubules bodies were present, and how they were arranged, still how they were formed, still the number of researches are going on. So if the centriole were present, then it is for the divided. It is divided into the two, and it resulted in the production of the production of the. They started the production of the microtubules or the spindle fibers. Then each pair migrates towards the respective pole. As soon as they were migrating towards their respective pole, and at the same time they were also started the formation of the spindle fibers or the microtubules. Here you can see the prophase second stage. Here it's very clear in this particular stage. You can see the two cells were there, and the chromosomes are totally dark in structure. There is no any kind of thing that the certain kind of thickening occurs or something like that. The chromosome shape and size will be the same as in case of the telophase second. Here you can see this is the stage of the ileum sepa, and you can see one half and two half. The proper cytokinesis occurs in this one, and you can see that this whole dark structure showing the chromosomes were scattered in the half of the cell, or you can say the complete half of the cell. That's why it takes the dark, the total dark color in the half. जो आपने उसमें देखा था टीलोफेस में, उसमें एक कोने में एक डार्क स्ट्रक्चर था पूरे सेल के अंदर। यहाँ पर ये फिर से ये पूरा का पूरा हाफ जो है वो डार्क हो गया है। तो दैट मीन्स कि क्रोमोसोम पूरे में वो फैल गए हैं। फैल गए हैं मीन्स कि आपका जो न्यूक्लियर एनवलप है और जो न्यूक्लियोलस है वो डिसॉल्व हो गए हैं, और जिसकी वजह से क्रोमोसोम न्यूक्लियर एनवलप से बाहर आ गए और वो पूरे में फैले हुए हैं, उन्होंने पूरे में स्टेन ले लिया है। देन come to the metaphase second. This is the metaphase second, and you can see how they were arranged with the help of the, as you know, that in phase one. Because all these things I am not going to repeat, because all these things I have discussed, you people, in case of the mitosis. So in the late prophase stage, what happens? The spindle fiber, they get attached to the centromere, or you can say that the kinetochore, uh, to kinetochore, so that the after the activity of the spindle fiber of the microtubules, they move towards their respective poles. Sorry, due to the due to that activity, they reaches to the equatorial plates. So on the equatorial plates, they look like that how they were arranged in case of the metaphase second. Now the chromosome organized as the equator and the equatorial plate, because most of the time, most of the persons, they use the equator words. That's why I'm using, but you must be very clear about that. That it's not an equator, it's an equatorial plate, because equator is something else. The centromere divided into two, give rise to the two daughter chromosomes. Everyone knows, nothing it is new, that and the centromere, what happens, the two sister chromatin, they were joined together with the help of the certain proteins that we will also discuss sometimes. Then each chromosome attached to the spindle at centromeric region towards their respective pole. Actually, they were attached to the centromeric region. They are not attached to the centromere. They were attached to the kinetochores. The two different protein bodies were present on either side of the sister chromatins. That I have also discussed when we were discussing about the mitosis. How they looks like at the metaphase stage, you can see that in this case, you can see that the two different stages were present. This is the one cell and this is the another cell. They were placed in a different way. ये सेल जो है वो एकदम जैसे आपको दिखेगा वो सीधा बुक में दिखता है, ये सीधा नहीं है, ये टेढ़ा है। ये हाफ है इसका सेल, और ये जो क्रोमोसोम्स हैं ये आपके मेटाफेस के ऊपर प्रेजेंट हैं। सो दैट मीन्स इक्वेटोरियल प्लेट के ऊपर ये मौजूद हैं। But at the same time, if you see this particular photograph and this particular stage, these are also metaphase stage. But you can see that they were not found at the equatorial plate region. They were not found at the equatorial plate region, and similarly, the case in this case and in that case. But as I said, that it's not like that whatsoever you have studied in your books, especially the whatsoever type of diagram which is included in your book. It's totally different whenever you are doing the experiments. So if you see this particular cell, this is the half one and this is the half one. In this case, it looks like that they were present at the equatorial plate, but in this case, it does not look like. As I said, in case in the mitosis classes, that suppose if you consider this is the one cell and this is the spherical cell, okay, and if you you can say that the chromosomes they were present at the equator यहाँ पर present है बीच में, ये पूरा सेल है और ये ऐसे करके यहाँ पर मौजूद हैं हमारे क्रोमोसोम्स इक्वेटोरियल प्लेट के ऊपर आ गए हैं। अगर आपके सेल की टैपिंग इधर से हुई, आपने आपका जो ये सेल है इसको इस तरीके से अगर टैप किया, तो आपके ये क्रोमोसोम यहीं के यहीं ऐसे आपको शो कर जाएंगे। तो ये यहाँ पर आपके मौजूद हैं, और एक स्लिट लाइक लगेगा, जैसे ये स्लिट है, एक डार्क लाइन टाइप की दिखेगी। वहां भी आपको बहुत एक एक क्रोमोसोम बहुत क्लियर नहीं दिखेगा। अगर आप बहुत ज्यादा एक्सपर्ट हैं, आपने बहुत अच्छी स्लाइड बनाई है, या फिर आपने कोई ऐसी स्पीसीज ली है जिसके क्रोमोसोम्स काफी थिक होते हैं, काफी मोटे होते हैं, काफी क्लियर होते हैं, और दूसरे कम होते हैं, तो वहां पर आपको क्रोमोसोम्स दिख सकते हैं। बट जो आपके पास माइक्रोस्कोप होता है, 10x 20x इवन 30 और 40x तक भी आपको ऐसा ही स्ट्रक्चर दिखेगा कि ये ऐसा अनयूजुअल सा कुछ स्ट्रक्चर दिख रहा है। सो अगर आपने इसको टैपिंग के जब की, तो वो सीधा आपने अगर इधर से कर दिया, तो वो आपको बिल्कुल ऐसा दिखेगा। अगर आपने टैपिंग दूसरी तरफ से हो गई, सपोज किसी भी पोल की तरफ से हो गई, या तो इस साइड से हो गई या इस साइड से हो गई, अगर आपकी टैपिंग, क्योंकि वो तो स्पेरिकल सेल है, उसके बीच में जितने भी हैं क्रोमोसोम्स वो बीच में मौजूद हैं, इक्वेटोरियल प्लेट के ऊपर पूरे प्लेट पे मौजूद है। तो अगर आपने ऊपर से टैपिंग कर दी, क्योंकि आपको तो पता ही नहीं है ओरिएंटेशन, ऊपर से अगर सेल की टैपिंग हो गई, तो इस तरह का स्ट्रक्चर दिखेगा, जिसको आप आइडेंटिफाई सर्विस बेसिस पे कर सकते हैं कि उसके क्रोमोसोम्स बहुत क्लियर और कंप्लीटली थिक होंगे। उसको आप कह सकते हैं कि ये मेटाफेज स्टेज है। So don't be confused with this particular figure, that they are not looking like the metaphase stage. They were not present at the equatorial plate. Anyways, this is about the metaphase. Now come to the anaphase. Anaphase also it's similar to the mitosis, as I said, that due to the help of the different enzymes, whatsoever type of proteins were present, they were dissolved, and these chromosomes or the sister chromatids, they were moved towards their respective poles. The two chromatids of the sister chromosomes were detached. Centromere separates completely and give rise to the daughter or the new chromosome. Daughter chromosome or the new chromosome. Daughter chromosome move towards the opposite pole due to the shortening of the chromosomal microtubules or the stretching of the interzonal microtubules of the spindle. So, by the activity of the microtubules, and also at the same time with the help of the different enzyme after the dissociation of the certain proteins which were present at the centromeric region, or which were helpful in attaching the two sister chromatids. So when they were moved, when they were these sister chromatids or the new chromosomes, they were detached. At the same time, the microtubules were also started to work, and they started to work and help in the movement of the chromosomes from equatorial plates towards the respective poles. So as soon as they were detached, as soon as they started to move towards their respective poles, the NFA secondary stage started, and it continue up to the cell reaches to the respective poles. Respective poles तक जब तक पहुंचेंगे तब तक वो interface second ही रहेगा. That I have discussed different times when we were discussing about the meiosis one and also the mitosis one. At this stage, the four group of the chromosomes were producing, having the haploid number of chromosomes. वहां पर हप्लॉइड हो ही गए थे आपके मेटाफेज फर्स्ट में ही. दो होमोलोगस क्रोमोसोम्स अलग हो गए थे दो डिफरेंट सेल्स में. अब क्या हुआ कि जो सिस्टर क्रोमेटिड्स थे वो भी अलग हो गए, और वो एक एक सेल को लेके एक एक सेट ऑफ द एक हैप्लॉइड सेट को लेके वो भी अलग हो गए, एंड दे वर फॉर्मिंग दी four different cells. Here you can see that in this particular photograph, certain kind of the projections were there in both the sides, and this showing that the chromosomes they were moving towards their respective pole. This is the diagrammatic representation. This is the original slide, and in this case, you can see the spindle fiber of the mitotubules were there, and they were responsible for helping the chromosome to reach the respective poles. Then come to the telophase second. In case of telophase second, the chromosome reaches to the opposite poles. Here you can see that the two cells were there, and they were moved to reaches to their respective poles. The nucleolus reappears, the nuclear membrane reappears. So this is very much important. The endoplasmic reticulum form the nuclear envelope around the chromosomes. Then the nucleolus reappear due to the synthesis of the ribosomal RNA and also due to the accumulation of the ribosomal proteins. These kind of activity and all these things. Just you can imagine. ये सब चीजें जब आप पढ़ रहे हो तो पढ़ते वक्त आपको बहुत सारी चीजें आपके जहन में आनी चाहिए कि नेचर आखिर है क्या कि एक एक बहुत ही माइन्यूट सी एंटिटी जिसको हम सेल कहते हैं, जिसको हम अपनी आंखों से नहीं देख सकते हैं, इतना छोटा होता है, उसके अंदर भी एक छोटा सा और स्ट्रक्चर होता है जिसको कहते हैं आप न्यूक्लियस. जस्ट इमेजिन लाइक दैट. क्योंकि आप जब एक्सपेरिमेंट करोगे तो आप देखोगे कि सेल कितना बड़ा है और उसके अंदर न्यूक्लियस कितना सा है, और न्यूक्लियस के अंदर भी न्यूक्लियोलस एक और एंटिटी मौजूद है. बहुत छोटा सा एक स्ट्रक्चर और मौजूद है जिसको कहते हैं न्यूक्लियोलस. और न्यूक्लियस के अंदर क्रोमोसोम्स जो है मौजूद है, और वो क्रोमोसोम्स जो है अगर वो अनकॉइल स्टेज में होते हैं, अगर वो आपके क्रोमेटिन नेटवर्क की फॉर्म में होते हैं, तो वो सेवरल मीटर्स के थिक हो, सेवरल मीटर्स की लेंथ होती है इनकी. जस्ट इमेजिन लाइक ऑल दीज थिंग्स. बिल्कुल आपको ये सोचने पे मजबूर होना चाहिए कि एक इतनी छोटी सी चीज, उसके अंदर एक कोई चीज जो मीटर्स में जो लंबाई है जिसकी वो मौजूद है, वो किस तरीके से कॉइल होती है, वो किस तरीके से उसका रेप्लिकेशन होता है, किस तरीके से उसका दो सेल्स बन रहे हैं, एंड ऑल दीज थिंग्स आर गोइंग साइमल्टेनियसली. कोई भी चीज उसमें ऐसी नहीं है कि अच्छा अब चलो अब ये हो गया अब ये होगा. जो होने वाला है वो सब ऑटोमेटिकली हो रहा है. अब इस एंजाइम की जरूरत है तो एंजाइम उसमें सेल में पहले से मौजूद है, बन चुका है वो एंजाइम. वो डिफरेंट प्रोटीन्स है जो आपने आपने पढ़ा होगा कि किस तरीके से जो है गोल्जी वेसिकल्स से प्रोटीन का मॉडिफिकेशन होता है, और वो मॉडिफाई करके वो पर्टिकुलर प्रोटीन बन जाती है जिसकी जरूरत होती है, और जैसे ही उसका काम हो जाएगा वो डिसॉल्व हो जाएगी. तो जस्ट इमेजिन कि नेचर ने कितना बारीकी से एक एक चीज को कितना बेहतरीन तरीके से ऑर्गेनाइज करके रखा है. तो नेचर इज द बेस्ट ऑर्गेनाइजर. आप इसको किसी भी लेवल पे डिनाई नहीं कर सकते हो. ये ठीक है, यू आर द मॉलिक्यूलर बायोलॉजिस्ट एंड यू कैन से दैट ये इसमें ऐसे हुआ, बट इमेजिन ऐसा हुआ क्यों. आप उसी टाइम पे प्रोटीन्स क्यों बनी, कोई जरूरी है जो है वो ठीक है आपने कहा ये पर्टिकुलर प्रोटीन बनी, ये बनेगी जब ये वाली स्टेज आई हुई होगी तो ये